If 0 people impacted by MS volunteered their bodies to do clinical trials, how many drugs would we currently have available to treat MS? The answer is 0. To learn more about MS clinical trials, don't turn away, because that starts right now. Howdy. Thanks for learning about MS with me, Aaron Boster. I started this YouTube channel to help my own MS clinic patients learn between visits, and it's my hope that through these videos, I can help you learn too. Today's topic, multiple sclerosis clinical trials. What are they? How do we conduct them? Who participates? And what do they teach us? Let's jump in. When I started in the field of MS, there were 3 therapies available. They were all shots. And I remember when I went to interview for my residency program to become a neurology resident, and I told the interviewer, a doctor that was asking me questions, that I wanted to go into MS. He said, "Are you going to go into research?" And I honestly answered, "I don't know yet." And he said, "Well, young man, let me share something with you. If you don't go into research, the best that you can do is peddle what's currently available, which in your field isn't very good. If, however, you participate in clinical research, you can help develop and bring new things to market to help your patients." And I think that's absolutely awesome. A good portion of my job as an MS neurologist is participating in clinical trials. And throughout my entire career, I'm very proud that I've participated in clinical trials to help bring new drugs and new diagnostic tests to market. It's a really exciting portion of my job, and it affords the people that I get to work with an opportunity to fight back, to go above and beyond simply treating their disease, to stick it to the disease, and to help future generations of people with MS have better and better options. I love that. Who participates in clinical trials? A manufacturer has developed a product, a hopeful drug, that they think is going to be the next best thing. And they're an active participant in this process. But they don't conduct the clinical trial. A second party comes in, called a CRO, or a clinical research organization. They're hired by the manufacturer to physically conduct the clinical trial experiment. But the CRO doesn't have any patients. And so they go out and they partner with clinical trial sites. A clinical trial site is very commonly large MS centers, whether that be in private or non-for-profit or academic, where there will be doctors and patients who are busy in the throes of taking care of clinical MS. And the CRO will partner with these clinical care sites to do the experiments. At the care site, there's a host of characters that will be involved. There's a primary investigator, and that's a doctor specializing in the field of MS who partners closely with the CRO and will oversee the clinical trial at that site. The primary investigator often serves the role as the treating physician. They are delivering clinical care in the context of the trial to people impacted by MS. The way clinical trials are constructed, you also require a second clinician. This is called a blinded evaluator. They're another clinician at the site, whether that be a doctor or a mid-level provider, and they are blind to what's going on with the patient. They're not directly involved in the patient's care. They don't talk to the patient about a relapse or a new symptom. They come in blind to what's going on with the patient, and they do formal neurological testing, neurological examinations, and they write down the scores. Having a treating physician and a blinded physician is a key element to clinical trials. It keeps us honest. If a patient told the treating physician, "Oh, I can't see," and then the treating physician did an exam, that will influence the treating physician's exam. So having a blinded examiner that's unaware of what's going on prevents that risk for bias. Also in the clinical trial, there needs to be a clinical trial coordinator. These are specially trained men and women that work with the primary investigator, with the blinded evaluator, and with the CRO company, and they sort of coordinate the clinical trial at that site. Now, the way clinical trials are constructed, the primary investigator needs a backup in case he's out or sick or on vacation. The blinded evaluator needs a backup in case they're sick or out on vacation. And typically, a clinical trial coordinator may have a second coordinator that helps out if need be. The most important person in the clinical trial, of course, is the human being impacted by MS. Think about this for a second. If people with MS weren't willing to volunteer their bodies to find out if medicines work, we would have zero drugs available. It's only by the grace of people with MS that are brave and willing to participate that we're able to bring new drugs and new therapies and hope to the field. And so the most important person involved in a clinical trial is the person with MS that says, I want to fight back. I want to stick it to the disease. And at a given clinical care site, the primary investigator will enroll people with MS that want to participate in the clinical trial. Now, depending on the specifics of the clinical trial, there's obviously other elements that are necessary. For example, most all clinical trials use an MRI machine as an imaging metric for what's going on. And so each clinical care site needs to have access to an MRI machine where there's a dedicated radiologist that's been trained in the protocols for the MRI and does them compliantly. Depending on the clinical trial, there will be a laboratory involved collecting certain blood samples or sometimes spinal fluid samples. And depending on the trial, there may be other clinical staff, such as an ophthalmologist to do special eye exams, or a dermatologist to do special skin exams, etc. All right, so we've introduced the cast of characters. We have a manufacturer that's made a product that they think is going to be awesome for treating MS. They have partnered with a clinical research organization, a CRO, that's gone out and found a bunch of clinical trial sites. Each one of those sites has a primary investigator doctor who's going to be the treating physician, a blinded evaluator that's going to evaluate the patients without knowing what's going on. They've got a clinical trial coordinator on the site that's coordinating the efforts that are going on at that site, and they enroll people with MS into the clinical trial. Now what? Ostensibly, what you're trying to do is you're trying to prove that the product that we want to study is better than something else. And we do that in the context of an experiment. So let me share with you how that breaks down. But first, a coffee break. That's really good. Okay, so when you do a clinical trial, you'll take a group of people with MS and you'll divide them up. Now, let's say that we divide them in half. So you'll have half the people over here and half the people over here. And you'll give all these people the drug of interest. We'll call that the study drug. And you give the other half the people something else. And then we're going to follow them as we move forward in time and see which group does better. And there's a lot of details involved to make sure that the clinical trial isn't biased and that the results really reflect this drug doing better or worse than this drug. So the first thing I want to talk about is called randomization. When a given patient agrees to participate in the trial, there's sort of a coin flip done where they're randomly assigned to either be in group A or in group B. They don't get to pick which group they're in. The primary investigator doesn't pick which group they're in. Nobody does. It's done randomly. And the patient isn't told which group they ended up in. That's called keeping the patient blind. Also, the clinical trial site, the primary investigator, the treating physician, all those people, they don't know which allocation they're in. And because both the patient is blinded to the allocation and the doctor is blinded to the allocation, that's called a double blind. So we have a double-blind clinical trial where we have randomly assigned people with MS to either be in Group A for the study drug or Group B in something else. Now, that something else could be many things. Back in the day, when I first started to do clinical trials, that something else was also oftentimes placebo. So placebo is a dummy drug or a sugar pill. It's not real. It doesn't have any active ingredients in it. In 2019, we don't do placebo-controlled trials in the United States for MS any longer. Why? Because there's over 19 drugs to treat MS, and it's not ethical to ask someone with multiple sclerosis to enroll in a clinical trial and on a coin flip end up on nothing. That's not okay. And so what we most commonly do in modern clinical trials is we randomize patients to study drug or standard of care drug. This is a real MS drug that's been proven to work by the American FDA. So we're pitting the study drug against something that we already know works. Now, the study design can be very, very complex, and I'm going to share with you one of my favorite study designs. It's called a double-blind, randomized, double-dummy active comparator. So that's a lot of words. So we've already talked about the randomization. Flip a coin, end up there or there. We've talked about the double-blind, meaning the patient and the doctor don't know which category the patient was enrolled in. The active comparator means that we're testing the study drug or a known active drug. Now we get into the concept of double dummy. If you were studying a pill and you experimented it against a shot, and half the people started to take a pill and half the people took a shot, well, very obviously you'd be able to figure out which was which. In order to hide the allocation, here's how you do it. Everyone who's on the study drug, and in this example we'll say it's a pill, they also take a shot, except the shot's fake. So they're taking a pill and they're taking a shot, a real pill and a fake shot. Over here, all the people that were randomly assigned to take the standard of care, the MS shot, they also take a pill, except it's a fake pill. They're taking a real shot and a fake pill. So everyone in the trial is taking a shot and a pill. Half of them are on active study drug in fake comparator. Half of them are on the real, half of them are on a comparator and fake study drug. And this is called double dummy. Now, as we follow people going forward in time, we call that a prospective evaluation. And typically in a clinical trial, the way that that's done is patients are brought in about every 3 months. And when they come into the clinic, we collect a lot of information. We ask them special questions on questionnaires called patient-reported outcome measures about a host of things. How are they feeling? How are they sleeping? What are their energy levels like? Are they depressed? Etc., etc. And that's a very formal way of collecting information. We do neurological examinations. The treating physician, the primary investigator, will do an exam, but that's not the one that counts. The blinded evaluator will come into the room that day and they'll tell the patient, hey, look, I'm not allowed to ask you how you're doing. I have to do a neurological examination without really talking or learning from you. So they do the exam, they write the answers down, and they leave. We also typically draw blood to do a myriad of various testing, sometimes to look at how the drug's working in the blood or to look at how the drug might influence the liver or the kidneys, etc. And oftentimes the MRI will be involved, maybe not every 3 months, but oftentimes every 6 months or 1 year. Depending on the clinical trial, there may be specialized eye tests, there may be visits by other specialized doctors like a gynecologist or a hematologist or a skin doctor, etc. And the patients will be seen at a cadence of about every 3 months, getting exams and collecting information and having MRIs every 6 months to every year. And this will typically go on for a period of 2 to 3 years. And this is a traditional sort of clinical trial design in the modern era for MS. So let's talk about the outcome measures, the things that you're trying to measure in the clinical trial. Before the trial starts, they clarify the things they're going to be looking for. And typically you'll have a primary outcome measure, a secondary outcome measure, a tertiary outcome measure, and an exploratory measure. So the primary outcome measure is the main thing you're trying to figure out. In most MS clinical trials, that's typically annualized relapse rate. What does that mean? So a relapse, a flare, an exacerbation, an attack, this is when someone with MS suffers from an acute bout of inflammation in the brain or spinal cord, which results in losing a neurological function. It typically comes on subacutely over hours to days, and that deficit will last for weeks to months and then get better. And I'll use, for an example, an optic neuritis, where someone loses vision in their eye and it hurts when they move their eye. And so we have two groups of patients. We have people that are receiving the study drug and people in the active comparator. And there's groups of people. And we look to see in this group how many people had attacks and how often. And in this group, how many people had attacks and how often. And we calculate the annualized relapse rate. so the frequency of attacks in a given year, and that's a number. And we can compare the number of relapses here, the annualized relapse rate, compared to the annualized relapse rate there. And we can do statistical measures to figure out, did this happen more often or less often than this? And is the difference in the number of relapses something that could occur just by chance, or is it less likely to occur by chance and it was actually because of the biologic effects of the drug? And so that's an example of a primary outcome measure, annualized relapse rate. Secondary outcome measures are other things of importance, but maybe not as important to the clinical trial as the primary outcome measure. And this could be things like confirmed disability progression. So when you're examining a person with MS in the trial and they develop a new deficit, again, I'll use this optic neuritis, and you can then see 3 or 6 months later if they got better, if that recovered or not. And if the person developed a deficit, they can't see very well, and 6 months later they still can't see very well, we'll say that they've confirmed their disability progression. And again, you can see if the group A had more or less confirmed disability progression compared to group B. And you can do statistics to figure out if that happened just by chance or if it was more likely a biologic effect of the two drugs that were studied. Another coffee break. That's really good. The MRI is oftentimes used as a secondary outcome measure. So we'll do an MRI at the start of the trial, and then we'll do an MRI at the end of the trial. And for each patient, we'll see, compared to the first MRI, did the last MRI have more spots? Whether that be contrast-enhancing spots or the T2 bright spots or other things. And we can see in group A, how many new spots on average did people get? And in group B, how many new spots on average did they get? And was the difference the same or was it different? And was the difference something that could just happen by chance or was it statistically significant? Tertiary outcome measures and exploratory outcome measures are typically things that we're interested in learning about, but we might not have the statistical power. We might have not modeled the trial to really answer the question definitively. It's stuff that we're going to learn more about. And this may be a great way of testing out a new concept in MS, like a new measure that we want to study, or a new patient-reported outcome measure, or a new blood test. It oftentimes is very interesting, but you wouldn't base the results of the trial on that exclusively. Now, traditional outcome measures in MS trials include things that I just talked about. The annualized relapse rate, so how often does a group have an attack on average in a given year? The confirmed disability progression, getting worse on exam that stays that way for 3 or 6 months, and traditional measures on the MRI, new contrast-enhancing lesions, or new or enlarged T2 lesions. There are newer measures that I find to be much more interesting and much more clinically relevant, which are now being increasingly incorporated into clinical trials. These include things like confirmed disability improvement. So instead of saying someone developed a deficit and it stayed that way, they're looking at someone that lost a deficit. Their exam improved and the improvement persisted for a period of 3 to 6 months. CDI, that's one of my favorites. We can also, in the context of the trial, look at NEDA, no evidence of disease activity. So given a period of time that the patient was studied, did they have nothing new happen? No new attacks, no new disability progression, no new spots on MRI, NEDA. And we can compare the two groups. On the MRI machine, we can look at brain volume. All people over the age of 18 have brains that are pre-programmed to shrink a little bit every year, but it's only a little bit, a very small amount. In the setting of multiple sclerosis, however, people can have accelerated brain shrinkage, sometimes 10 times faster than normal. And we can study whether a drug slows down the shrinkage. And we can look at an MRI at the beginning of the trial and then at the end, and we look at the rate of shrinkage, or how much brain shrinkage occurred. And we can see if the group on the study drug had more or less brain shrinkage on average compared to the active comparator. So that's a brain volume assessment. We also can look at provocative new laboratory tests. Many of you know that I'm fond of serum neurofilament light chain. Neurofilament light chain is a measure in the blood that looks at axonal loss or inflammation. It's really cool. And modern clinical trials are starting to measure blood levels of neurofilament light chain to see if they dropped or normalized in Group A compared to Group B. We can also look at things like patient-reported outcome measures and ask, did this group of patients report improvements in, say, depression more or less compared to this group? And so those are some of the newer clinical trial measures that we're seeing entered into modern clinical trials. If you're impacted by MS and you want to up your game, check out this playlist right there. YouTube thinks that you would love this video right there. And if you haven't yet subscribed to my channel, please consider doing so by clicking that little circle with my face in it. Go ahead, click my face. My name's Aaron Boster, and thank you for learning about MS with me. Until my next video or my next live stream, take care.